Dr. Sweetser, thank you so much for taking time out of day to speak to us. We really appreciate it. And I understand that you have discovered a new disease or something that might contribute to a previously undiagnosed condition. Is that correct?
Well, yes, it's not that we discovered the disease, but we undiscovered a very novel cause of this disease that gives us some insights that I think will have profound implications to the whole field of genetics. So,
Absolutely. And what was the condition that you were looking into?
so this specifically, maybe I can give you a little bit of a background about this case because this came to us in as part of our involvement in the Undiagnosed Disease Network. So this is a network of now about 15 hospitals around the country where physicians and geneticists can refer their most complex patients, and usually these are patients that have undergone all of the standard genetic testing that's available clinically and have not come up with answers. And so this was a boy, he was 16 years old that we saw him, and he had been had a very difficult course of his life, primarily multiple strokes that he had been having, and he'd been treated as part of what was considered to be a vasculitis, and but it wasn't a satisfying diagnosis in that he had other abnormalities that were going on, that he had some difficulty since birth, and so it turned out that this patient, as we put all the pieces together, he matched this clinical syndrome called smooth muscle dysfunction syndrome. And so when we thought we had the answer and we started looking for what the known cause of this gene, of this defect was, in all previous cases have had a very specific mutation in a gene called ACTA2. That's a smooth muscle actin. And when we searched, we did not find any mutation there. So this is where we put all the rest of the tools together that we have as part of the Undiagnosed Disease Network, including sequencing his entire genome, looking in all of his genomic data, and we came up with a very intriguing change that was appeared to be a mutation in a gene that was brand new, not seen in any of his parents. But it was a novel type of a gene. It was a gene called a microRNA that doesn't encode proteins. And so it's not exaggeration to say practically all of the genetic diseases that we really know the cause of right now are caused by mutations in protein coding genes. And one thing that has bedeviled us for many years is, even as we've applied all of the advanced genomic technologies to try to understand the causes of diseases, including sequencing all of the protein coding genes in the body, in many cases, we still can't come up with an answer. And we're wondering, what is there about the complexity of the human genome that can cause diseases that's subtle?
And so if this gene doesn't encode for proteins, what does it do?
What all the genes in our body are regulated. So there's a complex regulation that undergoes about how the genes are processed. They're all broken up into pieces and they have to be spliced into a transcript. And then they also have to be turned on and off at the right times. And helping us to do that are a number of nucleic acid molecules. So these aren't proteins per se, but these non-coding RNAs are essential for splicing correctly, turning genes on and turning genes off. And so in essence, they're almost like master regulatory genes.
So in the case of the young boy, what does your discovery mean for his treatment?
It does actually change the thought process about how they're treating the child. The whole treatment plan to try to reduce his strokes and try to treat him has really been centered on trying to reduce inflammation, and that does not, although there might be a small component of that, that doesn't seem to be the driving force behind all of his problems. With these types of genes, there is the possibility of designing therapies that can replace these dysfunctional microRNAs. So there is sort of the possibility that we might be able to use this information for designing new treatments for this disorder in this patient in particular.
Absolutely. So it would have implications for gene therapy.
Yeah.
Once we get down that road and
Down that road, yeah.
turn it into a pill that you can take and just replace the faulty gene. That'll be
Yeah. Just as an aside, it's kind of interesting you mentioned that, because we're starting another clinical trial with a genetic disorder. So there's a metabolic disorder where your body, there's so many of these disorders where your body is missing an enzyme that it needs to process certain foods and can cause disease. And the first metabolic disorder that people might know as PKU, phenylketonuria.
Yes.
And so we're starting we're one of the sites testing a trial sponsored by Synlogics, where it is a pill. It's actually a bacteria that's been engineered to produce an enzyme that the body's missing. So it is actually popping a pill to replace the enzyme, so you don't have to worry about changing your diet.
And what did it feel like when you found this gene and you figured out that this might really well be the cause?
Well, this is one of the first cases that we've had of a microRNA that we've discovered as part of the Undiagnosed Disease Network, and I've been involved for nearly 9 years as part of this, working up 100 patients or so, and we've really made our focus, as everybody else has, about looking at these protein coding genes, just because there were not very many examples of these non-coding genes causing disease. And so the discovery of this really helps kind of open our eyes as validating that these types of genes can cause diseases. And so as we go searching more and more to the cases that remain undiagnosed to us, it gives us something to go back and look toward to see if there might be a discovery waiting to be made.
Well, this has been incredible. Thank you so much for taking time of day to speak to us. We really appreciate it. And best of luck with all of the future research after this. It's going to be really exciting.
Okay, thank you. Yeah, I welcome the chance to share it with the audience here. Thank you very much for the opportunity.